Technical Veterinary Learning TVL channel, founded and produced by Professor Ahmed Mamdouh El Sharif, Professor of Epidemiology and Infectious Diseases of Animals, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University. Viral diseases of camels. Rift Valley fever in camels. Rift Valley fever is an acute arthropod-borne viral disease. Was first reported in Kenya in 1930 and has since made irregular outbreaks in small ruminants and cattle, with associated zoonotic spread to humans in sub-Saharan Africa and the Arabian Peninsula. Camels usually have an inapparent infection with the Rift Valley fever virus, but sudden mortality, neonatal mortality, and abortion occurs, and abortion rates can be high as in cattle. This released by OIE at 2018. Outbreaks of Rift Valley fever are rigidly connected with heavy rains and flooding that increase habitat susceptibility for arthropod vector populations. Diagnosis. Rift Valley fever may be suspected based on clinical signs characterized by a storm of abortions, insect activity, and concurrent disease in small ruminants, cattle, or humans. Epidemiology of Rift Valley fever disease. Rift Valley fever virus is a negative sense tri-segmented RNA virus that belongs to the genus Felibovirus, family Phelonioviridae, order of Buonaviridae. Rift Valley fever, Felibovirion and genome. The realm is Riboviria. Kingdom Orthonaviri. Phylum Nigarnaviricota. Class Eloviricites. Order Buonaviridae. Family Phenoviridae. Genus Felibovirus. Species Rift Valley fever Felibovirus. There is only one serotype recognized, but strains exists for variable virulence. Phylogenetic analysis of the genome of isolates from camels in Mauritania in 2010 suggested a shared ancestor between the Mauritania 2010 strain and strains from Zimbabwe, Kenya, South Africa, Uganda, and the other strains linked to the 1987 outbreak of Rift Valley fever in Mauritania. This figure shows Rift Valley fever virus under electron microscope. This is a diagrammatic molecular aspect of Rift Valley fever virus virion, where we will speak about viral protein functions. Gn and Ge are important for viral attachment and entry. The envelope glycoprotein Gn is responsible for requirement of the virus protein to the Golgi apparatus. Gn is also independently involved in the packaging of nucleus into virions. The glycoproteins are also involved in efficient viral release from infected cells, for which the primary stimulus is the inscripted genome. The nucleus protein encapsulated genome RNA by protein RNA and protein interaction with form of the R nucleoprotein. The L protein forms part of the RN protein core and plays a visual role in the primary transcription of messenger RNA transcribed from the viral genome soon after viral entry and the secrets of the genomic RNA replication in the cytoplasm. G, N, G, E, and nucleus proteins make up the major antigenic proteins and are therefore the common targets for diagnostic and vaccine development. The non-structural proteins NSM 78 kilo data, GNSM and NSS are associated with virulence. Both NSM and the 78 kilo data protein were shown to be dispensable for viral replication. However, it was observed that infection of Culex with Rift Valley fever virus lacking the 78 kilodalton displayed reduced transmissivity and infection rate, whereas it was completely unable to infect Aedes aegypti. Nucleus protein is targeted to the mitochondrial membrane and plays a role in delaying the onset of virus-induced apoptosis. Nucleus protein is also dispensable for the viral life cycle, but its major virulence factor due to its ability to suppress host antiviral responses. Nucleus protein is localized at both cytoplasm and the nucleus, where it forms filamentous structure. One of the earliest known functions of it is ability to inhibit interferon function of the cell. It was later found this protein specificity blocks transcription from the interferon P promoter by interacting with it. Genome organization of Rift Valley fever virus. The encoded open reading frame are shown as solid black arrows on the large L, medium M, and small s segments of the viral genome. Untranslated region UTR flank each segment. The length of each genomic segment is indicated in kilobases. Transcript messenger RNAs are depicted as dashes arrow with the start codons AUG indicated as black boxes, and the resulting proteins shown as shaded boxes. Proteins encoded by the M segment are produced by post-translational cleavage of translated polyproteins. The nucleoproteins is generated by an messenger RNA transcribed from the negative sense genomic viral RNA. The non-structural S protein is represented on the genome sense sRNA as a white box and is produced from an messenger RNA transcribed from the antigenome copy of virus RNA generated during viral replication. The octagon represents an intergenic transcription termination site. Rift Valley fever virus is susceptible to various disinfectants such as ether, chloroform, sodium deoxycholate, formalin, sodium or calcium hypochlorite. Geographical distribution of Rift Valley fever disease. According to OIE, Terrestrial manual, camels have been regularly involved in Rift Valley fever epidemics in East Africa, Egypt, and more recently in Mauritania. Rift Valley fever disease emerged in Egypt in 1977, in Mauritania 1998, and in the Arabian Peninsula in the millennium, and repeated in Kenya in 2006 and 2007. Countries that reported outbreaks of Rift Valley fever disease in camels include Mauritania, Egypt, Sudan, and Kenya. This is the map of countries reported outbreaks of camel Rift Valley fever, taking red colors. Risk factors include above the average rainfalls, floods, and human cases in the same area. Additionally, camels are suspected of playing a major role in the spread of Rift Valley fever disease from northern of Sudan to southern of Egypt in 1977. This map showing the areas of Rift Valley fever outbreaks in Egypt since 1977 up to 2003, where the disease from the southern part of Egypt, from Aswan up to the Delta governorates are distributed. The disease transmitted from animals to human and vice versa through Calyx and Aedes aegypti. This map showing the temporal distribution of Rift Valley fever from 2006 up to 2009 in Africa, where the disease taken of South Africa, Madagascar, Kenya, and Sudan, in addition to Yemen. This map showing the temporal distribution of Rift Valley fever from 2009 and 2010, where the disease distributed in Mauritania and Senegal, then Botswana, South Africa, Madagascar, Kenya, south of Somalia, and Sudan, Djibouti, and Yemen. Distribution of Rift Valley fever from 2011 up to 2012, where the disease distributed in Botswana, south of Africa, Madagascar, Kenya, and south of Somalia, Sudan, Djibouti, and Senegal, Nigeria, and Mauritania. Temporal distribution of Rift Valley fever from 2013 up to 2015, where the disease reported in Mauritania, Senegal, Nigeria, Sudan, Kenya, Uganda, Botswana, South Africa, Madagascar, Zimbabwe. Temporal distribution of Rift Valley fever during 2016 and 2017, the disease reported in Mauritania, Senegal, Nigeria, and Côte d'Ivoire, Uganda, Botswana, Zimbabwe, South Africa, Madagascar, Kenya, south of Somalia, and Sudan, in addition to Niger, Mali, and border of Chad. Temporal distribution of Rift Valley fever during 2018 and 2019, where the disease reported in Mauritania, Senegal, Niger, Côte d'Ivoire, and then Uganda, Chad, Mali, Sudan, Kenya, south of Somalia, Madagascar, Botswana, Zimbabwe, Uganda, and South Africa. This map showing the distribution of Rift Valley fever in Africa. Blue denote the countries with endemic disease and substantial outbreak of Rift Valley fever. Green color taking the country known to have some cases, periodic isolation of the virus, or serological evidence of Rift Valley fever. CDC in this map taking in consideration historical information on human and animal cases, as well as the detection of Rift Valley fever antibodies with different serological tests. They classified the countries according to the epidemiological situation of Rift Valley fever in 3 classes. The endemic one taking the blue color, the countries showing sporadic presence taking the green color, and the unknown status taking the light color as it presented in this map. Animal susceptibility. Age has been shown to be a significant factor in the animal's susceptibility to Rift Valley disease. Rift Valley disease in camels is secondary to infection of small ruminants with the same virus, with few outbreaks in which obvious clinical signs are seen in camels. The camels were added to the list of susceptible animal species when infection was reported in dromedaries for the first time during the Rift Valley fever outbreaks in northeastern Kenya at 1962. Serological evidence of dromedary camel infection with Rift Valley fever and the demonstration of specific IgG antibodies in camels indicates that these animals are naturally infected. In most of outbreaks, Rift Valley fever disease also involved camels besides sheep, goats, and humans, with abortion as the only clinical symptom. More recently, 34% of seropositivity and showed that Tunisian 100 dromedaries camel succumbed to the disease and may contribute to its dissemination among farmers and other livestock. Mode of transmission of Rift Valley fever. Rift Valley fever is transmitted by insect vectors to direct contact with organs or fluids of infected animals. Rift Valley fever virus doesn't cause persistent infection or carrier status, and certain Aedes species act as reservoir for Rift Valley fever virus during inter-epidemic period. Several different species of mosquitoes are competent vector for the Rift Valley fever virus, primarily Culex species and floodwater breeding Aedes species that transmit the virus from infected animals to other animals and to humans. If we look to the Rift Valley fever virus ecology, there is 3 parts: enzootic cycle, epizootic cycle, and spillover to human being. In the enzootic cycle, maintained via transfer from parent mosquito to offspring, then local and low-level transfer of the disease to livestock, and this happens during periods of average rainfall. While the epizootic epidemic cycle, it is amplified transmission, where heavy rainfall and flooding enable Aedes mosquito breeding environments to flourish, and then enzootic outbreaks cause abortion storms in animals with 90% mortality in newborns and 10 to 30% mortality in adults. Secondary vector, including other mosquitoes genera, such as Culex, can pass on the virus to human and animals. Spill over to humans, exposure to blood and tissue of infected livestock occurs during slaughter or birthing activity. Humans can also be infected with Rift Valley fever via bites of infected mosquitoes. Pathogenesis. Until 2019, there are few information known about Rift Valley fever pathogenicity, but after that date, the Rift Valley fever virus infection shows unique pathogenesis in each animal model experimental infection. Because viral replication and host antiviral responses most probably contribute to viral pathogenicity, an understanding of Rift Valley fever pathogenesis requires identification and characterization of the viral virulence factor and host antiviral factors. This figure showing the molecular pathogenesis of Rift Valley fever virus, where there are 9 steps for the Rift Valley fever virus from entering the cell until forming the new progeny of the virus. Step number 1, virus attachment and entry, and then endosomal fusion between the virus and the endosomes inside the infected cell. Number 3, RNA proteins released into the cytosol, where this leads to transcription of the virus and translation of the RNA virus by the nucleus. Then replication of the Rift Valley fever virus occur. Then recurrent to Golgi apparatus, where the virus make packing of its variants. The last and 9 steps is budding and release of variants. This is the progeny of the new viruses. Clinical picture. Generally, the incubation period of Rift Valley fever varies from 1 to 6 days and could be less than 24 hours in newborn animals. Clinical disease is usually seldom seen in adult camels, but pair acute infection with sudden deaths, abortion, and some early postnatal deaths have been observed, as said by Scott et al. 1963. In 2010, scientists reported confirmatory evidence for a field camel infection with Rift Valley fever virus in northern of Mauritania, involving mass abortions in small ruminants and dromedary camels, and at least 63 human clinical cases, including 13 deaths. The clinical signs of Rift Valley fever disease varying depending upon species of animals, age, the physiological status of animals affected. In camels, clinical signs other than abortions were reported in this animal species, including hemorrhagic sepsemia and severe respiratory distress. During the outbreak of Rift Valley fever in Mauritania, 2 clinical forms were manifested in dromedary camels as follows. Number 1, a pair acute form with abrupt deaths in less than 24 hours. An acute form showing fever, ataxia, ballooning, edema at the base of the neck, audible expiratory wheeze, and ventral positional dyspnea, blood-tinged nasal discharge, icterus, severe conjunctivitis with ocular discharge and blindness, hemorrhages of gums and tongue, foot lesions, nervous symptoms, and abortions. This picture of a dromedary infected camel with Rift Valley fever showing conjunctivitis and ocular discharge, hemorrhages of the gums, and edema of the tongue. A figure of hemorrhages of gums and tongue in an infected dromedary camel with Rift Valley fever. Foot lesions were cracks in the sole of the infected camel due to Rift Valley fever with presence of secondary myiasis. Edema at the base of neck of an infected dromedary camel infected with Rift Valley fever. Dead camel with signs of abortion, convulsions, and arching of the neck. Laboratory diagnosis. Specimens for laboratory diagnosis are hazards and must be handled with extreme care. Samples includes: number 1, from live camels, samples are blood in edita, clotted blood, plasma, or serum. Number 2, from dead camels and or aborted fetuses, samples are tissue samples of liver, spleen, kidney, lymph node, heart, blood, and brain. The most common laboratory procedures includes: number 1, reverse transcriptase polymerase chain reaction RT-PCR. This figure shows multiplex RT-PCR amplification for Rift Valley fever virus and others, where Rift Valley fever virus here are present in the 5-lane Rift Valley fever virus strain Smith-Pern. IgG and IgM antibody enzyme-linked immunosorbent assay ELISA test are used for detection of Rift Valley fever virus. Virus isolation by cell culture require high containment laboratory. Virus detection by electron microscope, as we see the arrow showed this transmission electron micrograph despite a highly magnified view of a tissue that had been infected with Rift Valley fever virus with 131 kilobyte magnification. Differential diagnosis. Rift Valley fever disease have to be differentiated from brucellosis, enterotoxemia, PPR in ruminants, anthrax, and hemorrhagic septicemia disease. Zoonotic capability. Rift Valley fever is a viral zoonotic disease that primarily affects animals with the capability to infect humans. In humans, the disease takes different forms ranging from mild to severe and fatal forms. The mild form is characterized by a flu-like syndrome with feverish signs, muscle and joint pain, headache, and loss of appetite. Severe forms include severe hemorrhagic fever, ocular form, and meningoencephalitic form. People at risk for Rift Valley fever disease include herders, veterinarians, and slaughterhouse workers. Virus transmission occurs in humans conducting veterinary procedures and when handling infected animals and meat via wound from an infected knife, contact with broken skin, or inhalation of aerosols produced during the slaughter. the documented index case linked to sick camel. The country's government reported the outbreak located in Kenya, Wajir area in the northeast to the WHO on June 8, 2021. This case began on June 2 when an 18-year-old man who is thought to be the index case patient was seen at local health facility with a 5-day history of fever, weakness, bleeding, and other symptoms. He died the day he was hospitalized, and an investigation found he had reportedly eaten meat from sick camel toward the end of May. 2 days after the man was hospitalized, 2 of his relatives were hospitalized also with similar symptoms, including a 25-year-old man who died on the day he was admitted. Blood samples from the 2 patients were tested at the Kenya Medical Research Institute, where one of the samples was positive for Rift Valley fever virus presence. As of June 8, 10 suspected cases have been reported, including 5 deaths. 3 people have recovered and has been leave the hospitals, and 1 patient is still hospitalized. Testing on 8 samples is underway to Kenya Research Institute, according to the WHO, and the samples were positive for Rift Valley fever virus. Treatment of Rift Valley fever disease. There is no specific treatment for Rift Valley fever in camels. Prevention of Rift Valley fever disease. Measures applied in the face of an outbreak include the following steps. Number 1, control of animal movements. Number 2, control at slaughterhouses surveillance within containment and or protection zone. Number 3, quarantine measures application. Number 4, vector surveillance and control. Number 5, ante and post-mortem inspection. Number 6, vaccination by several types of modified live attenuated and inactivated virus vaccine of Rift Valley fever, but little is known about their use and efficiency in camels. Notification. Rift Valley fever is an OIE enlisted disease, and immediate notification of clinical cases upon detection to OIE is mandatory. Final important conclusions on Rift Valley fever disease in all warm-blooded beings. Since the first recognition of Rift Valley fever disease in an outbreak in Kenya at 1930, more than 92 years has passed and showed good progress in characterizing the clinical, pathological, virological features of Rift Valley fever virus infection. But Rift Valley fever virus still causes outbreaks in African countries or the Arabian Peninsula. The development of effective, safe, highly immunogenic, and economical vaccines for animals and human beings will prevent Rift Valley fever disease in the endemic country. There are several important questions to address in controlling the Rift Valley fever virus and in further understanding its pathogenesis. Some of these questions are: mechanism that triggers hemorrhagic fever. Number 2, route of viral entry to the brain. Number 3. Mechanism of prolonged diseases in Rift Valley fever patients and animals in the presence of neutralizing antibodies. Significance of vaccination to prevent Rift Valley fever after aerosol exposure and infected insect bite transmission. Number 5. Biological weapon. Rift Valley fever was one of more than a dozen agents that the United States and other countries researched as potential biological weapons before the nation suspended its biological weapon program in 1969, but there is no documented information about this suspended because of information opacity all over the world. These addressing questions will have a substantial impact on understanding of Rift Valley fever pathogenesis and development of anti-Rift Valley fever virus reagents in the future. We're keeping you on the right track, so please subscribe the channel to reach to the most newest knowledge about the epidemiology and different infectious diseases of different species of animals. See you later.